So today I'm going to talk about the concept of mitosis. So mitosis is a very, very fundamental topic in cell biology. However, most teachers just fail to get the message across. Personally, I've had 4 or 5 different classes where the teachers have tried to teach me about mitosis, but I never picked anything up from him. However, I've actually established a way to get things done and really get it to sink in. So the first thing that you got to do is you got to think about what has to happen during this process. In order for a cell to divide, what are the things that need to get done? Well, in my mind, there are 3 major components. There are 3 major steps. So the first one, you got to replicate the DNA. So for each chromosome, you got to make another copy of it. Because remember, you want each cell that arises from it, each daughter cell, which there's 2, to have the same number of chromosomes, and for those to, I guess, be identical to the parent cell which it aroused from. Number 2, we gotta split those chromosomes. And then number 3, finally, we gotta split the cell. We gotta go from 1 cell to 2 cells. So now what we're gonna do, we're gonna just draw this out real quick. So let's take a look at what's happening here on the cellular level, the microscopic level. So first thing we gotta do, so we start off with our cell, and then essentially what's happening is you're replicating everything. So here, this is just all the DNA. It looks like a mishmash, a pot of soup, per se, because everything's just being copied. Then, from there, we gotta split everything. So let's have everything splitting. So we take our copies, and now we're just gonna, now we're just gonna split them. Make it a little nicer. So what's happening during the splitting is that you're going to have 2 sides, or sort of 2 poles to the south. This is your first pole, and this is your second pole. You know how you have the north, this is like the north pole, and this is the south pole, and here's the equator. That's a good way of thinking about it. So all the DNA, or all the chromosomes, are starting at the equator. So identical copies are each going to be, each of the identical copies are going to be pulled to the opposite pole. So one's going to be pulled to the north pole, one's going to be pulled to the south pole. And finally, what we got to do is we got to actually split what we have here. So we got this nice cell, and here's the other cell that's going to arise from it. And let's just crop this out nicely so you really get a good view of this. Boom. And then we're going to have these chromosomes, I guess, sort of ending here and ending here. It's not really going to look like that. So we had 3 things happen here. We had the replicate, we had the split, and then we had the second split, splitting of the chromosomes. So one chromosome copy went to one end, one chromosome copy went to the other end, and then we had splitting of the actual memory of the cell. It just so happens that these 3 things I just described are actually major steps of the process of mitosis. The first one is interphase, which was the copying of the DNA. The second one was anaphase, which is splitting the identical copies, putting one in each daughter cell. And then the third one was telophase, which is splitting the cells themselves. So it just so happens that these are 3 of the steps out of the 5 total steps that there are in mitosis. So there's a good acronym for that I came up with here. So everyone says, oh, it's IPMAT. Okay, but how are you going to remember that? It's just a general acronym: interphase, prophase, metaphase, anaphase, telophase. Well, there's 2 ways to think about it. If you just get rid of the I, okay, I comes first, whatever. PMAT, you could think of it as sort of a standardized test. You have the GMAT, the LCAT, the LSAT, the MCAT, and then we have the PMAT. So this is a standardized test for mitosis, or even better, IPMAT. You've taken a biology class, a very general basic biology class. I'm sure your teacher has talked about the central dogma of cell biology. So the central dogma is DNA to RNA to proteins. Well, think of the word dogma. Think of what a little dog does. A dog will pee on the mat. So I pee mat. There. I pee mat. Now you're going to remember. I pee mat, central dogma mitosis. It's too stupid to forget. So let's really get back into it. So first, let me just move these over. Over here. So let's look, what are we missing here? So we're actually missing, after interphase, we got prophase and we have metaphase. So let's just draw out what's happening during prophase. So prophase, so think about what happens when we're going from interphase to anaphase, what happened right here. So first of all, we got rid of this nice nuclear membrane, which is sort of like the skull covering the brain, because we consider the DNA or the brain, and we have to split the brain after we copied it. Another thing that happened is we actually had a physical mechanism pulling the copies of the identical chromosomes to opposite ends of the cell. So we got to set up that process, of course. So the first real setting up step, or the real big setting up step actually, occurs during prophase. So nucleus here. So three things happen during prophase. First, I guess the most negligible one, the DNA that we replicate here starts getting a little more organized. So the chromosomes start organizing themselves a bit. They become, I guess, little straight things rather than whatever the hell you want to call that. Then second, this membrane we have here starts breaking down. Because think, in order to actually split these apart, you have to get rid of that membrane here. Let's get rid of that a bit. Boom. So we call it the nuclear membrane, or some say the nuclear lamina. It gets broken down. And third, this is something I didn't include here. We have these little, as we call, centrosomes. So we have these centrosomes. These are sort of like the pulleys or the cranes that really get the work done. These are the workhorses of the process in my mind. So what happens is during this phase, the prophase, you have to have these centrosomes move to opposite ends. So let me just mark this step. Boom, let's put that here. So this was prophase. So we just went from interphase. Oh, is that over? Oops. Interphase to prophase. Nice. So now we went from moving these guys over, getting rid of this nuclear lamina, still disorganized, so we gotta get from prophase to anaphase. So this is the spot. This is the, I guess, phase that most people tend to be most familiar with. This is the process of, should I just move everything over a bit? I don't know where those came from. Get rid of them. So next step is going to be metaphase. Everyone knows about metaphase. For some reason, that's the only step anyone's ever heard of, metaphase. So let's see. During metaphase, this is where things really get lined up. This is where things really get set going. Good enough? Good enough. So what happens here is we're going to really set up the two poles. So we have the identical chromosomes lining up. Then at each pole we got our centrosomes now. And then from the centrosomes, we're going to get our, let's do this nicely here. We're going to get what our microtubules coming out. So essentially these are more or less rigid poles that really come out from the foundation here. This is our foundation, or the pole, and then we got a crane, or it's like a tow truck. Call it a tow truck that has all these lines coming out of it. It's got all these lines, and we're going to be towing all of these sort of, like a simultaneous manner. It won't really be simultaneous, but it's pretty close to simultaneous. So we're going to be pulling those to the opposite ends. Just mark this here. So this is our step of metaphase. So we're going to, some may call this the metaphase plate. We got our two poles here, and actually because I put these here, I might as well put them on the other, in anaphase as well. look at this. I'll just make it orange. That's what I want to do. Oops. Here we go. Here we go. And that's that. Okay. I'm not an artist here. And then I'm just going to add arrows to clarify things. Clarify the direction here. Black. Another one. Another one. And that's the process of mitosis here. So let's look the whole thing over. So essentially, first, interphase, we replicate. Actually, first, let's go over 3 steps. So we have to replicate the DNA. When we replicated the DNA, that was interphase. Then we had to split the chromosomes, that was anaphase. And we had to split the cell, that was telophase. So let's look at the end product of telophase. Some like to say, oh, it looks like this in the end. So I guess we got our two sort of daughter cells. I don't know why they call them daughter cells. Could be daughter cells, son cells, child cells, little cells. We always describe them as little, but in the long run, I guess they're supposed to sort of be the same size as the other ones. Just put that in the middle here. Oops. There we go. So let's look the whole thing over again. So going from interphase, anaphase, telophase, replicate, split, chromosome, split the cell. And then between that, we have to do prophase and metaphase. This is to really set things up. So prophase was responsible for, I'm going to write that out actually. It's right at the 3 things what happened during prophase. So I guess the chromosomes became a bit more organized. Then we had the, I guess, nuclear membrane degraded a bit. Oops. And lastly, we had the centrosomes migrate to opposite poles. And yeah, so there's a reason why this whole process is so important and why teachers emphasize it so much, because it's actually a very critical piece in cancer. So what happens in a lot of cancers is this whole cycle just happens uncontrollably. As a result, you want to stop some of these things. So for example, in one of my later videos, I'm going to talk about drugs such as Taxol, paclitaxel, whatever you want to call it, that actually stabilize these microtubules. So because they stabilize them, this process can't happen normally. Remember, if you have them stuck at the equator with these parallel chromosomes lined up and you can't divide, this whole thing just gets messed up. What will happen in the end is either somehow you'll end up reverting back to this state, sort of some say G0 state, or you'll end up getting all these blebs going in different directions because, okay, you'll pull some things here, you'll pull some things there, and then it's just a messy situation where the cells can't survive because they don't have the proper number of copies. So this is the whole process, and hope you got something out of it. Just remember PMAT, or IPMAT. What does a dog do? Central dogma of biology. It's what a dog does. A dog pees on the mat. So IPMAT. It's too stupid to forget.